To present my work here. So, as the title suggests, I will talk about the role of the SIK tyrosine kinase in breast cancer cells. And this is joint work between a theoretical biophysics group in University of Montpellier and a cancer research group in Montpellier as well. So first, what is SIK? SIK is the spleen tyrosine kinase. It's a very well-studied tyrosine kinase in immune cells. But it has also been shown to have an oncosuppressive role in breast cancer. In more details, it has been observed that there is a higher number of metastases happening in absence of SIK than in presence of SIK. And its expression is indeed reduced in the most aggressive cancer cell lines. So it has been considered as a bad prognosis marker for breast cancer cell. But very little is known about the mechanism behind that. Incidentally, most tyrosine kinases are usually considered as being pro-oncogenic, so the case of SIK is slightly surprising. And SIK itself has also been observed as pro-oncogenic in other types of cancer, especially lymphoma. And SIK inhibitors are being proposed for lymphoma and some autoimmune diseases. So the fact that that inhibiting SIK might increase the risk in case of breast cancer may have an important therapeutic role. So one of the questions that we wonder here is what makes SIK special compared to other tyrosine kinases, and can we identify some of the mechanism behind this surprising role in breast cancer? So very quickly, we will do some bioinformatics for that. So starting with a list of target proteins which are differentially phosphorylated in presence or absence of SIK. We identify some enriched pathway, build some large network of known interaction between many proteins in this pathway, and selected some subpart of this large network, which is the part that I will mostly talk about now. So first, where does the data come from? We use the SILAC method, which enables to do differential proteomics. So we have both SIK positive and SIK negative cells. And we enrich the proteins that are phosphorylated on tyrosine and identify peptides using a mass spectrometer. And we started from two already available data sets comparing breast cancer cell line in presence or absence of SIK. So one was done in the MCF7 cell line, which is naturally SIK positive. So they use the SIK inhibitor for the comparison. And the other is MDA231 cell line that is naturally SIK negative, so they reintroduce SIK in this cell line. So these two data sets have roughly 250 and next to 500 identified proteins and very little overlap. Yet, if we look at the enriched pathway, we find very similar pathway in both cases. So we decided to study the full list of target protein together. And this enriched pathway, they cover cell adhesion and cytoskeleton related mechanism, which fits pretty well with the fact that we know that SIK is involved in the aggressiveness of the cancer cells. So based on this list of pathway, we selected a lot of this enriched pathway, and we built one of this very popular hairball that you've probably seen thousands of them yet. And in there, we'll start searching for some path connecting SIK to the target, to the identified target. And one of the examples, looking for just the shortest path between SIK to the Cortactin protein, which is a known regulator of the actin cytoskeleton. So this enables the identification of 7 possible paths, which are the shortest path in terms of topology in this network. But looking at that, we have 3 of the protein in this candidate path that have not been identified in our data set. So maybe we should consider that they are less likely to be the relevant one in this case. Yet, if we look at the gene ontology data to identify the modifier of tyrosine phosphorylation, this small subnetwork contains only 2 tyrosine kinases. One is SIK itself, and the other is SARC. So SARC is the only protein that is upstream of Cortactin in this network that is also a tyrosine kinase, which makes it a lot more likely to be the actual predecessor. And so we decided to go a bit further than very simple topological shortest path and to add some custom weight to the links. In particular, we decided that it was slightly better if we could reuse identified protein from the dataset along the path. And we also considered that it was much better to go through phosphotyrosine modifiers in the step before the identified proteins. So using this weight, now we have only two candidates, two remaining main candidate paths between SIC and Cortactin, and they both go through the SARC tyrosine kinase. And indeed, it was experimentally validated that SARC is required for the differential phosphorylation of Cortactin by SIC. So it's not so clear on the Western blot here, but you can see that a bit better on the quantification. In absence of SIC, there is no phosphorylation of this of either SARC or Cortactin. In presence of SIC stimulation, then you see high phosphorylation level. If you remove SIC, it's strongly decreased. And if you remove SARC, we keep the phosphorylation of SIC, but phosphorylation of both SARC and Cortactin is diminished. So another case study is a case of Ezrin, which is involved in adhesion and migration. And here in this new set of paths, we, in the step right before Ezrin, there is no good candidates. There is no tyrosine phosphorylation modifier. And indeed, if we look a bit closer at the data, it happens that some of the previous work shows that there might be a direct link between SIC and Ezrin. In particular, the second dataset that we used, Eliuk, did a follow-up study where they showed that that SIC directly phosphorylates one of the peptine residue in Ezrin, in vitro. And actually, the very same residue has been identified in vivo as differentially phosphorylated in presence of SIC. And the two proteins have also been shown to colocalize. So we propose that in this case, the effect of SIC on Ezrin is a direct one. So stepping back a bit, we can show the effect of adding the weight in the search for shortest path. So this is an overview showing the connection between SIC and the identified targets that are associated to adhesion and motility, still in gene ontology. And the blue nodes and the blue links are the ones that we only find if we do topological shortest path. The white ones are the ones that we found using topological shortest path and we keep when adding weight. And the orange one are the ones that are introduced by putting back the weight. So we didn't see that in the example network before, but it also happens that in some case we will select some longer paths because they are more mechanistically relevant. Yet, this study of shortest path is still a bit crude. In particular, as we have a very small range of weights that we can put on the interaction, we often come up with a large set of candidates mechanism, and then we don't really know where to start experimental validation. So we need to discriminate between several mechanistically relevant paths. And also, if we discriminate too much, we will come up with a single proposal, and then the biologists will want to do what to try next when this fails. So the aim of what comes next is to do both of these refinements and to come up with a list of alternative candidates that can be ranked. So the first thing that we did for that is to refine the weight by using random walk. So we start with a network with integer weights that are based on our biological criteria to begin with. And then we turn this weight, which has some kind of cost, into probabilities by inversing them. Then we perform a random walk with restart, starting from SIC, and we use that to assess new probabilities associated to the transitions. and we turn them back into weight to refine the search for shortest path. So this will give us a much wider distribution of the weight in this network. Then, again, a very similar network than the one I showed before. The blue nodes are the one we add with the weighted shortest path, the simple one, and that will vanish after adding the refinement through random walk. And we also introduced some paths that were first considered as longer in orange again. So the next step is how to select not only one best shortest path, but to keep several of them. And for that, a very popular method is to use the K shortest path, which consists in selecting a threshold K, and instead of picking the shortest path, we will select the K shortest path. It's very popular, but I see two problems with that. First, I mean, it's pretty much the same problem. The K plus 1's shortest path may be just as good as the K's one, and but it might also be a very bad one. So there is no real way to pick a case that will work out good for everyone. So instead, we propose to use near shortest path. That is also an existing method, but that is less used in this area. This one consists in selecting a K that is a factor that we apply to the weight. So we compute the weight of the length of the shortest path, and we will consider, for example, that we keep all the paths that are not more than 20% longer than the shortest one. So we implemented that with a modification of the classical Dijkstra algorithm. Yet we don't have yet experimental validation of this result. So more work is needed to check if it actually works in practice. So here we have a method to explore existing knowledge, select some hypotheses, and to come up with a ranked list of alternative candidate paths. And I want to highlight the fact that it's very important to select well the weight that we will put on the interaction, and to make it fit well to the data set from which we start our study, and to mechanistic criteria. And one interesting note is that we cannot obviously predict missing link with that. Yet we showed in the case of Ezrin that we can still point inconsistencies which might help to identify some of these missing links. Yet, of course, inference methods are much more powerful in this case when we are not lucky enough to have a direct one. And as a side note, my main area of interest is the dynamical models. And so I see this work as a first step toward building mechanistic dynamical models. If you want more details, it has been recently published in PLOS Computational Biology. Question? Yeah.
SILAC data usually give you not a single protein, but rather protein groups, where several proteins which have the same peptide, for example, are considered together. How would you recommend, whether you look at this problem, and how would you recommend to deal with this kind of situation when the same thing can be separated into many?
Yeah. So the question was on the use of SILAC data, which doesn't always allow to. In SILAC, we identify peptides, and then we have to map them on proteins. Some of the peptides can be mapped on a unique protein, some can be mapped on several, and there is a lot. So I didn't really talk about the SILAC analysis itself. In this case, we started from the list that was published, and so pretty much most of the workflow for the SILAC analysis data, the peptides that are not uniquely mapped to a protein, that they are often associated to the most likely one based on the specific peptides, which is not so convincing. And indeed, there is a lot to be done on SILAC analysis itself here. So what I showed here is really considering that we have a good starting list, but SILAC analysis data has a lot of problems, indeed.